Thanks for interest in that. So I want to talk a little bit about how we use generative AI mostly to model cellular state, a little bit about response, but then also sort of to get a little bit interpretation in. And I think this idea, this revolution, I mean, I think in our field in computation, I think generally in computation biology, we've been using machine learning for a long time, right? But being generative about it, I think it's sort of interesting. Kind of ties a little bit into excitingly what Jimmy was showing before, that you sort of want to add statistical inference, really get sort of maybe variational error bounds, things like that to whatever you do. And this is old saying from Feynman, famous physicist. They said, like, what I cannot create, I cannot or do not understand. This sort of, you have this forward physics-based models where you understand things. And you could argue that if you generate something, you do create, but people would maybe more call this black box. So I think it's really interesting back and forth between these two aspects. And I think maybe for our purposes, often it could be a good model of a cell that sort of, if it behaves well under perturbations, you have a prediction. Is that maybe where you want to go? Or do you want to have sort of really detailed pathway mapping? I think both sides can have an argument for it. I think it's interesting where this will hold. This is from an older review we did with Nicholas Rejewski, this picture of cells going through this high dimensional state space, going through various decisions if they sort of take a wrong turn, I think is really exciting. I think a big part of our research in single cell biology, I think, would be to learn these cellular landscapes and then to be able to act upon it. That's kind of what you could call generating, right? So you want to sort of see what happens when your cells maybe sort of make this trajectory, then you can use trajectory inference for that. Maybe what happens if, sort of cell takes a wrong turn, goes into one particular disease state, can you maybe then sort of understand this landscape part, and can you maybe act by sort of finding the ideal perturbation to go back? And, these perturbation scales, they're actually, these assays that allow you to do high throughput perturbations also on single cell RNA-seq level, which is sort of what we mostly focus on, I think might enable us to address some of these questions now. Before I start, I just want to sort of really briefly say that engineering matters. My lab has been developing ScanPy for analysis, single cell analysis in Python. And because, to really maintain a growing ecosystem, we realized we need to really partner up with colleagues and friends, where we set up this SCVerse consortium, which now has a whole bunch of shared data structure, but a whole bunch of core packages. And then basically the Python single cell ecosystem around that sort of interacts with these packages. I really invite you to contribute or work with us if you're interested in this. We recently had the first SCVerse conference, really very nice growing community, very international. And I think it's sort of exciting directions coming in there. For example, now scaling everything up, our 2.0 release next spring, summer will be to sort of run a ScanPy on at least 1 billion cells. I think these scales are coming up, but then you get everything GPU accelerated. So these engineering aspects are important. We've been doing things for spatial analysis, particularly also a multimodal spatial framework together with Olli Stiegler's lab. I think important sort of baseline sorry for that, baseline data structures to then build upon with potential machine learning models. We've been recently releasing PerPy, a perturbation package where you can sort of also visualize then perturbation effects in sort of a one level up, sort of in perturbation space, which you can sort of see as a population representation. And also been talking about how to do these sort of how to do analysis. So this is just part on the engineering aspect, which so many of us I think are dealing with to get things useful. But I want to talk about this action on these cell landscapes. And with my intro, you might have seen already that a beautiful review from Croatia and Peter Sorgo some time ago. They called this differential biology. But there's this sort of this back and forth between these principle-based models that we, in biology, because of all our priors, often take, and then the data-driven ones. And this may be sort of the hybrid way to go, are they back and forth. And I want to share with you that we can maybe take some data-driven models and make them a bit more principle-based, or at least interpretable. The type of framework that we use, of course, is going to be deep learning. I think with the scales that we see, I think this is somewhat inescapable. These type of autoencoders in our field that sort of take these gene time cell matrices, then learn them in an unsupervised fashion, were first introduced by Yusuf and my lab sort of in parallel, where essentially sort of minimize the reconstruction loss, and then you sort of get this sort of cleaned up data in particular, but you get this bottleneck layer that's sort of interpretable. You can add conditional autoencoders where you then sort of give that information, thereby sort of removing sort of being able for this network to use this information, thereby sort of in this bottleneck, removing this batch effect, sort of have this most efficient encoding. And that's a very useful technique for batch correction, data integration, and so on. It gives you then these transitions, for example. And here's an example of really just a two-dimensional bottleneck for PBMC data, where you see that they really pretty much look like a t-SNE plot, or UMAP plot. So they really identify the core structure in the data. I think it's very exciting. So then, you want to interpret these latent spaces. I think what's important is that these bottlenecks, though, like each dimension in itself doesn't mean much. They just sort of encode the data. But by adding priors, sort of, going from this purely data-based one to maybe a hybrid one, you can start interpreting it, adding perturbation effects, things like that on. And if you want, in a very weak fashion, just even telling it about batch effects is sort of some prior knowledge, right? We, together with a bunch of colleagues, have been running this data integration competition at NeurIPS, and we've been actually doing this now up to now, also with perturbations and so on. And we see that these things are quickly taken up sort of leading ranks just because they're so flexible. And I recently read from Pass Bio, I think they've been saying that nowadays a single paper has more than maybe 500,000 cells or so in as sort of a new contribution. So these scales are also getting big. So I think it's going to be useful to take up these flexible models. But where's this generative? Do you guys remember this picture of the Pope in the funny jacket? I've been showing this a while, but this is an example of generative AI, which is not actually really. It's not real, right? But how has it been generated? Well, because there's real pictures of men with white coats, there's real pictures of men, and if you sort of take the difference of these, you get this white coat, you add them, so you generate them. This is sort of how these generative picture models work. How can we use these generative effects in our field? Well, this was an idea that we've been sort of proposing some time ago, where you take now a cell under a perturbation that could be sort of maybe something that you measure, right? And then you subtract the ground state, and then you sort of have this new set of cell lines where you want to sort of extrapolate where this effect would go. And this is then how you can also be generative about it. So you can sort of do this latent space arithmetics visualization that I showed you before. Also in single cell RNA-seq space, we initially proposed that it works to some extent, but of course, you sort of need to do more to get this to work. How is that principled idea? I wanted to talk about perturbation effects. I've been mostly speaking about this gene expression space in a minute, but I just wanted to review this part briefly. If you see transitions of cells in a few examples, let's say this could be different cell types, what we do is then for a new one, if you want to predict that perturbation behavior, just as I showed you with this latent space arithmetics, you encode them, you go this latent space. In this latent space, then you expect that these things turn linear. That's kind of what we did with this addition effect. And if you then have a new one, you apply this perturbation, you go back. Of course, there's a big assumption, right? Why would they be linear? Computer vision, in some examples have shown that this actually makes sense. Often nowadays, we want to actually put additional priors in to make that happen, right? And of course, then we can sort of predict this. So in a few extensions, in particular also one with Stefan Günnemann and one with Meta AI, we've been extending this to add sort of embeddings of these deltas. So this would be, for example, a drug space or also CRISPR space, but then also making sure that you get these linear transitions. And I think the most recent one where we work with optimal transport type of constraints is a very exciting way to go. And we're recently combining this with diffusion-based models that are sort of the most expressive versions in computer vision to really make this happen. I won't be showing these results, so this is basically the ongoing stuff, but I think this is already useful. But what you will learn in many of these examples is that they generate and sort of behave well for letting sort of supporting maybe an experimental design, take it the next step. But this out of distribution generalization, this is sort of the key task that you want. You want to sort of be able to go into a new, let's say, new direction that you haven't been measuring. That's hard and really needs massive data. And there could be an argument that we might not be there yet in single cell perturbations. And already in that early paper, we found sort of linear methods to be performing well in CPA. We benchmarked this as well. And of course, you also don't model the dynamics, right? Remember, we had this Waddington's landscape idea, like how do you see that here? So I think there's room for extension. Just to summarize this part briefly, if you want to model a cell's response to a drug, right, you can use these generative AI techniques to extrapolate this response profile. But we have all this information. Central dogma could be one, right? Like, how can we add that information? And for me, one of the most beautiful examples how that works, sort of these really prior systems biology type of approaches in our field is RNA velocity, as has been initially introduced by Stelinissen and Peter Kachenko a few years ago. This idea being that you have this splicing metabolism that you now measure, and you sort of assume it's independent in that simple system across all genes, and then you just write up a very basic differential equation. But because it's decoupled, you can actually solve this in closed form. And then if you sort of plot unspliced versus spliced, you sort of get this dynamical behavior, right? So can you combine this with these latent space techniques, right? I mean, because, with this one, you sort of estimate rates, you embed them, and then you sort of get this sort of vector field, right? Very, very useful thing. And recently with Nir Yosef, we've been actually combining this sort of estimation technique here with a way how to put this into a sort of combined perturbation space where you can then sort of see where these transitions, not just embed, but where you can sort of also get an uncertainty model for that. Still, this assumption that these things are decoupled are there. So it's not, so it gives you sort of this dynamic behavior, but it sort of doesn't give you the wiring under Waddington's transfer, as you might have seen. These are like an underlying GRN or things that push that direction. In parallel, there have been a whole bunch of techniques that deal with the idea of learning regulatory networks instead. So not just doing this, but actually sort of seeing where a perturbation would go. There's, for example, Cell Oracle from Sam's place. I think she was talking earlier. Dynamo from John Weissman, Pando from Barbara Treutlands. There, we've been doing cell rank sort of interpretation of these dynamical models. But these things work well in terms of sort of getting the wiring in, often from single cell attack or also databases. But the true dynamical nature is not really interpreted. So you don't get the dynamics. So you have this RNA velocity, you have this regulatory network, so can you sort of combine? This is sort of the idea what I want to show you in as sort of one of the first things we've been doing. So here, what you can do is you can write out the dynamics of sort of the full transcriptome state in a cell as of a function of its previous state, right? And that could be, of course, just G expression, and then this F somehow needs to encode the dynamics. This is, of course, challenging. That's why, people usually don't do it, because like fully estimating that means estimating a 10,000 dimensional space across like hundreds of thousands or millions of samples nowadays. So that's why we usually assume this thing is decoupled, right? But we think that we can put these things now together if we assume a regulatory prior, and we call this RegVelo. Let me show you briefly the idea how this works. This is, by the way, work of really talented PhD student Wei Shu Wang, who has been working on this for last 2 years. So what this does is it bases itself on that embedding model that you saw before from this collaboration with Nir. So, but in the same time, we also assume a prior regulatory graph, right? So this prior graph we then use to constrain it. For these unspliced splice matches, we do the typical encoding decoding. We get a latent state, and then from the prior graph, we know basically for each transcript, based on these edges that we sort of take from the graph, a particular level of transcription rate. This transcription rate level we then put into the dynamics, in the dynamic model that you saw before, but now we couple it. And we actually also have a sort of soft version that sort of allows us to extend this graph beyond what we just originally have. So altogether, we get this regulatory kinetics, and then we can write down a likelihood for a particular set of observations and then fit the data. The key problem is, of course, the parameter inference. And in this case, we just use variation inference. But because of this sort of high dimensions, sort of the key contribution, I think, would say that instead of now fitting 10,000 one-dimensional ODs, which actually you have a closed form, you now have this coupled one. So you need to solve this on the fly. And the trick is that you have these very flexible neural network type of inference models that can basically reduce this to a neural ODE and then solve this. So this was really the major achievement, I think. So let me show you what this thing can actually do. So for example, here you now see a single cell a model where we actually have a label for cell cycle just with a Fuji marker, right? And we can now see how the dynamics of RegVelo look like, and they kind of seem to be sort of correlating nice with the marker, but we want to be quantitative about it because we want to sort of compare with the non-regulatory network prior based models. So for this, we measure things such as cross-boundary correctness, where you sort of go through the cell cycle phases, some confidence in the velocities estimated in correlation with the known prior. And then we see that we get sort of robust values here. We can compare them in a minute to other methods. Similarly, because we don't only estimate dynamics, but we also estimate a regulatory network. We sort of adapt the given one, right? We can ask how well that sort of fits to known sort of states in that cell cycle dynamics. And we can sort of look at the edge predictions and particular weight prediction models. And if you now compare this to existing models, just for the velocity side first, you see that this rate-value approach outperforms with respect to a whole bunch of scores, this inference, consistency of velocities, and also the latent time. And the edges you sort of now can compare to models such as Cell Oracle, or what we showed before. Using the dynamics essentially helps it to sort of get also better edges. So it's sort of this constraint based on regulation, but allowing it to smoothly extend it, allows it to build both better networks, but also because of that constraint, inference more dynamic, more robust dynamics. Really excited about that. We can do this now. The beauty now, of course, is if you have an underlying sort of more prior-based model, you could be much more robust in going out of distribution. Remember before I said in these purely data-based ones, based on, let's say, diffusion models or so, we need a massive amount of data to be OD. But here now we have a prior, so we can just basically take a network and sort of mask some type of regulon, right? And then simulate the dynamics, look at the local effects, and then we can see how the original dynamic looks versus the perturbed one. So we just look at the perturbation versus the original one. And then you can sort of look at the effect of particular perturbations. So let's try this. Here we do this for an example from pancreatic endocrinogenesis, where you have here progenitor cells that then go through sort of early pancreas development and then form different cell types in the islet, such as beta cells, alpha, and then also delta and epsilon cells. And by just sort of looking at the dynamics of the vector field that rate-value estimates, but then applying a cell rank on top of it, you get the terminal states as you would expect them. So that works very well. And we can actually also show that this sort of number of terminal states that we get sort of works much more robust than compared to sort of our previous approaches of VELO and SCVELO. But in addition, you can now do knockout simulations. So you can take this one and you sort of do a knockout, for example, of NeuroD2, and then you see sort of that the epsilon cells don't exist anymore as is known before. And then you can sort of look into that screening of this and can say, what type of targets are actually mattering, and you sort of look at these particular ones, and can you do, for example, type of double knockout, and you actually see that you sort of most of them get alpha cells. And these things can be confirmed from literature. So that's, I think, quite useful for us. Let's now really look into a data set where you have sort of measured perturbations. In this case, we're really lucky to pair up with Tatiana and Siwan from Oxford, who have been looking into one system for quite some time, namely the zebrafish neural crest development, where they have a time course, both SmartSeq2, so very highly quality resolved full transcripts, as well as 10x multi-ohm cross time points of this developmental system. In addition, they've been able to set up also a transcription factor perturbation setup, where they can directly capture then in a droplet-based model these knockouts. So it's essentially like an in vivo perturb-seq. So first we can learn latent time. So you see UAP, I don't show you the annotation, but because we have different stages, we can sort of see that this learned latent time from rate-value nicely associates with the staging. So the latent time sort of gets grow up with this, and it's typically expected that they often grow wider, that there's some cells that will stay more early here. You can do terminal state identification, and then you get sort of hit missing pigment cells and so on as neural crest cells sort of then need to go in development. And you can show that you actually identify these sort of terminal states much more robustly than with previous approaches. But now you can do the perturbation, right? So you have a pseudotime, you have a model of regulatory dynamics, but now you can do perturbation. For example, here we look into a few driver genes for missing time, and then we see that if we do various knockouts, you actually get sort of an enrichment of particular cells and depletion of, for example, hit missing time, as would know if you kill those missing time driver. And you can do the same thing for pigment cell drivers, and then you sort of see a knockdown in the pigment ones. So you sort of get the right knockdowns as you would expect from literature. But because now we have this perturb-seq, right, we can actually also really look into the whole set of new factors that Tatjana was just hypothesizing. And what we see there is that in such a perturbation benchmark, we, for example, find that our rate velo, and there's sort of two versions of it. We have a soft one that sort of allows you to learn additional edges, and a hard threshold one really gives you much nicer correlation with the observed perturbations than dynamos or cell oracle or something like that. And this is consistent with a single knockdown as a combination knockdown. So just to summarize this, with this rate velo, we kind of have this hybrid model that takes prior information from a regulatory setting, like from a gene regulatory network or something like that. And typically, as you say, we use, I think now mostly scenic plus for inference from either given attack or previous one, and from a dynamic one by combining with velocities. Of course, there's a bunch of limitations still. The underlying regulatory dynamics are still quite simplified. So we have an additive sigmoidal model for that. So there's, I think, a whole opportunity for including epigenetic modifications. And I think adding additional modalities, of course, is going to be challenging. So to summarize this, how to model a cell's perturbation dynamics, I think it's really useful to write up a forward model. And with these powerful frameworks, we can actually also now do inference on this. So we combine this deep cell representation with these generative network. The second, and this is unpublished, so we hope to bring this out in the next few months. The second unpublished thing that I wanted to share is a representation learning. So yeah, obviously we want to learn atlases, right? And we've been doing this in human cell atlas. And one sort of example that I want to show you in a minute, where I think we can find better representations, work by Lisa and Malte in a big collaboration with the whole Human Lung Cell Atlas network, which is the Human Lung Cell Atlas. We've initially taken a bunch of data sets, harmonized annotation and metadata, anatomical location, and build these core atlas that with transfer learning, then we extended to, I think, something like 400 individuals, 2 million cells. So that was a really useful sort of resource for us that, in terms of data integration, we just used, I think, SCVI or ScanVI at the time. So essentially these conditional variation autoencoders that I was showing you before. So just sort of these pure conditional autoencoders. But it's useful. You can do all kinds of things on top of that, right? Because now you have many subjects. You can do association of cell types with particular downstream markers, patient representation learning, things like that. And we do find interesting things because you can map disease on top of it. We find, for example, a shared disease state in monocyte-derived macrophages where this factor SVP1 is high. That's actually shared across pulmonary fibrosis, COVID-19, and also some type of carcinoma. So I think that's actually interesting. We've been doing this now at scale together with the Human Cell Atlas for other organs, not just for the lung, but also for the pancreas, particularly for the islets at the moment. We're doing this for the whole pancreas. It's currently ongoing with Rui Chan's lab for the human retina, and most recently with Barbara Treutlein and Greg Camp's lab for brain-derived organoids. So we're sort of always doing this type of integration work. But you get then these individual dimensions, and these are going to be hard to interpret. So how can we do a hybrid model for that as well? So there's I think that's really a challenge that maybe some of us have been addressing by basically taking linear models, but often has been ignored. So you have these gene time cell matrices, and for most of the downstream analysis, such as clustering, integration, and so on, we want to have some type of latent representation. This used to be, for a very long time, this KNN single cell graph. With increasing complexity and scale of the samples, often it's been sort of replaced by a latent projection. And this latent projection could be something linear, but often is then these sort of new network or KNN integrated or whatever type of intermediates, right? So that's where we use these autoencoders, but then this thing turns non-interpretable anymore. It's kind of hard to go back to the original genes, right? So you always need to do this sort of index-wise. For individual ones you can do, but for gene programs it might be a bit more challenging. So these latent spaces are crucial, but they're difficult to interpret. And the solutions now to make them is that you either add really biological priors or just purely statistical ones. And I think it's important to say that this general question of finding a nonlinear function that gives you an optimal independent, it's often called disentangled meaning that if you sort of change this one variable in ground truth, only sort of one thing changes. This disentanglement is not possible if you don't add these inductive biases. So you need to do something like that. And there's various approaches. The purely data-driven ones could be, but maybe could you add also principal ones? So early on, for example, we sort of assumed some type of sort of decorrelation in that gene expression in the cell cycle space, which is wanted to take out known factors such as cell cycle. Neil, in a very nice perspective, and in many downstream applications, has been sort of proposing just doing factor decompositions. And your examples such as MOF are very popular, particularly multimodal settings, where you can sort of find the factors. So you sort of essentially find some type of independent decorrelated or something like that expression. So you do sort of fancy versions of PCA or IC or something like that. Other approaches are to add actually biological priors. Oh no, okay, no, there's some nonlinear ones. Sorry, I missed it. So there's we've been doing a workshop paper where we add some type of constraints to the VAEs. So some type of statistical constraint in the VAE, and we did a recent thing where we add actually gene program priors in the decorrelation, so you have a linear decoder. But these are all kind of, I mean, they work, they give you some latent space, but you always pay a price. So what we now asked is, can we do this disentangled representations where, you know, find this particular dimension where, let's say, one cell type sort of is sorted out, or you sort of find a dynamic process? Can we do this sort of a bit more systematically? And here I want to show you results from Amir, PhD student in the lab, who has been sort of hitting his head against that problem for some time, but I think found a really exciting idea. And here we build upon something that's called additive decoders that has been proposed at NeurIPS just a year ago, where there's really some nice theory on actually showing that you can do disentanglement. Essentially, the idea comes from computer vision, where they have this idea of sort of having this additive scene. You know, sort of you put like you put a chair in the picture, and then you sort of have this additive composition of scenes, right? But then these effects often are non-linear on the pixel space. So you have this additive scene, but you have sort of this non-additive behavior. So the latent space is additive, but the pixels, so the observed space, is something non-additive. And how can you bring these things together? In a sense, this is sort of what we want to do with gene programs, right? So this is the idea of the model. We take our typical variational autoencoder, conditional variational autoencoder that I showed you before. So you take the variables, encode them, you decode them again. But now we take this additive decoder. So we take the latent representation, but now we cut them in each of these individual dimensions. This could be univariate, or it could be multivariate. We add the condition to that, and then we individually decode them. So we have a nonlinear decoder here, so a nonlinear function, but just on much smaller dimension, typically one dimensional plus covariate. And then you get each of these representations. You do a lot of these decoders, and then you combine them with a pooling function. And here you do something nonlinear. For example, log X type of pooling. And what we can show is that these what we call pooled additive decoders, that actually then enable really general disentanglement. So this additive decoder disentanglement theory we can sort of pull up to this scale. And that's really exciting. That gives us also some theoretical grounding that we can actually do this now. So we use log sum pooling for this. And I just show you now that this actually works. So in this case, we take this long atlas that I was showing you before, and we just look at the embedding. And first, it's important to say that they're pretty much as expressive as before. So the latent space, you find the same thing, and the integration scores are pretty much the same as for SCVI. But you can now look into individual dimensions. And just on this UMAP, I plot you now a few of those dimensions where they sort of localize in this sort of cell type arranged space. And for example, here, okay, we find that here there's, for example, one particular cell type in there. This would be dimension 17, right? And if I sort of just show you a few interesting dimensions that are now really disentangled, here you find a process that sort of seems to be enriching in some sort of set of T cells, and a shared process. And then you also see some type of developmental dynamics here. If you plot those two against each other, you actually see that there's some sort of erythroid development happening. How can we quantify this more consistently? Well, because now we have individual dimensions, we can actually sort of play around with these, right? And this is the idea how we really make them interpretable. We sort of traverse not just one dimension, so we change one dimension and see what the effect in the gene expression are, right? So we just correlate them with the genes, or we identify them also relevant genes, okay? And then you sort of get the gene programs, things like that out. So this is what we now do for these various factors that I showed you before. And then you get a bunch of transcripts out that are, for example, here markers for dendritic cells, for Treg cells, you know, for some type of stress program and so on. And if I now just superimpose this here on these pictures that I showed before, is, oh yeah, here we pick up these MDCs. Here we sort of see this Treg substate. This shared process here is actually this association that's sort of seen all over. And you know, these two here would be actually markers for erythroid progenitors and for erythropoiesis. So you really get this latent space with pretty much similar integration quality, but now with sort of each axis meaning something. let me try to convince you that this is actually really systematically happening. And we have some theoretical grounding, but things in practice at scale and these high dimensions could always go wrong. So what we do now here is that we take now a ground truth variables and we just look at changes in one representation. And this is what we do by trying to formulate some type of ground truth. How can we do this? Well, let's say one ground truth could be known cell types. That's what I showed you sort of a bit before. Another ground truth could be if you attach particular perturbation, you could see where they go. Or maybe you have some underlying process. And then you want this to be only present in one of these latent dimensions. That's kind of what we measure. For this, we come up with some type of scores. And this is what I visualize you here now, right? And I compare, we call it DRVI for disentangled representation, variation inference, or DocDVI is sometimes called. And there's an additive pooling version of that. We mostly use this one to sort of previous approaches, linear ones, as well as nonlinear embeddings. And what you now see is that you always look at the average gain over this most simple linear PCA model. And then you see that DRVI sort of always outperforms across a bunch of data sets, a developmental one, perturbation one, and various batch ones at scale. Won't show you details too much on this, but I just want to show you that this actually works also in the lung. Before this was just a PBMC example, sorry for that. So if we do this here now in the lung, we get basically all these various factors out. So I plot you now here the different cell types, and I plot you which dimension sort of picks up the cell types. And you see that you really get pretty much dimension resolved for those various cell types, and then you get some sort of shared programs. So you really get now these interpretable axes in, and you can look into what's this shared one here. And for example, you see this one is sort of giving you some type of dissociated stress response, one myeloid one, and so on. The latest point I want to make is that now that you have a method that sort of finds individual dimensions, then you can ask, like, how much does it depend on the scale of the latent space? Like, is there maybe even like a sort of lower dimensional space that you would want to look in this whole thing altogether? And that's actually what we observe. So if we now increase, so here just we take latent space size 32, we see that, the size of the component at some point drops just at the end. But if we go to a larger one, it seems to be dropping again a bit later. So it needed that capacity, but drops. And if you go to 128, sort of it takes a little bit more, but it starts dropping again. And this is for the immune data set. But we kind of think there might be maybe some sort of maximal intrinsic data dimension or something like that in there. And this is actually what we see if we do this across those data sets that we benchmarked before. We see that there seems to be sort of going up, but at some point then thresholding the number of dimensions that's being used. And, this is starkly different from PCA or ICA or so, where you always sort of add additional sort of low variance components you keep adding. Here sort of really starts dropping. I think exciting phenomenon, and we're currently studying this. So to summarize this, I think in order to build interpretable latent space, potentially also atlases, we build them by integration of single cell profiles. And this we do with conditional variation autoencoders, but with DRVI, we can do so in a more interpretable fashion. So if we now have the atlas done, what do we do with these, right? So using those latent spaces, I think, becomes very important. And I think for this sort of like transfer learning, I think is the way to go, where you sort of have now your given atlas and you superimpose, you map sort of cell states on top of this, right? And we've been coming up with different ways how to do this. I think maybe quite exciting one is, and I just wanted to briefly advertise this here and mention, if you now have patient like sample level covariates, this could be like a disease classifier. This could be actually also something like QTLs or so. Like, this could be something like SNPs or so, where you can then call QTLs, which, typically are shared across all these cells. What you can do is you can do the aggregation across the individual cells using an attention layer. And we've been sort of setting this up here. We encourage you to check this out, or we're happy to chat about it. What I just want to wrap my talk with, though, is this extension towards these sort of really large scale models at the end. I was mentioning generative AI, and of course, the big kid on the block at the moment is this extension towards these large scale models called foundation models. And I just want to sort of put this a bit in context, because I think this idea of acting on these cellular landscapes is a really old one. In systems biology, this has been asked 20 years ago by these things, what they at the time called whole cell models. I'm sure you've seen at some point. They sort of, with the Human Genome Project being done, the hope was now we have all the bits and pieces, we can sort of put this together, put structure and everything, and then we build this very big thing. But then the predictions looked really poor, and just doing this in a purely first principle version kind of didn't work. And even though this has been sort of put forward, I think in principle these things were not actionable, and it's kind of kind of failed. But there's renewed interest. We have these data sets. We have these different modalities that can measure at the same time. So, Dimas Hasabe some time ago also mentioned, he wants to do a virtual cell in the next 10 years as one of his big dreams. CZI has been putting forward, particularly Mark Zuckerberg and Priscilla Chan have been putting forward to build this virtual cell modeling system as a thing to sort of lead to new breakthroughs. So I think this idea, putting these things together, is becoming really exciting again. And here again, we can use, in principle, this is, given now these latent spaces that we do more and more across atlases, I think it's actually a good setting, right? So what you would do is you sort of do some training on these large-scale samples, maybe masking of particular genes so that you find this embedding, or you take the evolutionary autoencoder ones that we showed before, and then you do fine-tuning for these downstream tasks. So this is sort of the key idea. Here we just put out a review of existing methods. I think the seminal one that really people that really got excited about is from Christina here, where you take single-cell RNA-seq, you learn in a self-supervised fashion a transform, and then you downstream fine-tune it for cell-level tasks such as cell annotation in particular, but then also for gene function, things like that. I think at the moment the vote is still out how well this actually generalizes and is sort of really outperforming simpler, linear, potentially more constrained models. But I think that the vision of such sort of train them once across very large scales, I think, is very exciting. We have a model where we actually add spatial constraint as an additional sort of harder fine-tuning task, where we think these things such as cell clustering, cell annotations is too simple a task where you won't be outperforming linear models in many cases. And then actually we see that if we do this really at scale, in this case at scale really means for us training across more than 100 million cells, which was really hard from all kind of engineering perspectives, so we don't even go into. We can do such tasks such as niche prediction, where you give us a gene expression profile just of a single cell, and you predict its local niche composition. And this is actually, of course, in a fine-tuned version for various organs. Very excited about this direction. Just wanted to flash this here, and I'm really looking forward to seeing how these new directions develop. Why? Well, we said we want to map things on top, and this is sort of mapping on top, like building like an actionable atlas, so to speak. So once you have these models, I think enabling then this lab in the loop, where you hypothesize like some new things, you test them, and you sort of go back. I think it's going to be really exciting. So these agents that then sort of act, we will maybe sort of support us in our research. Yeah, that's maybe one of the visions to go. So with this, and we've been playing around with this, and I think we have a really exciting solution, not just for bulk averages, but for sort of phenotype prediction that I encourage you to check out. Really happy to chat about it, but this is a bit off topic, so I've been not talking too much about that yet. We call it PROPHET, the predictor phenotypes. So with this, let me conclude. With generative AI, we can learn cell atlases and we predict perturbation effects. And I showed you two new approaches that we've been doing. One is called RigVelo, where we do dynamic modeling with gene-related network constraints. And then we can really do this sort of dynamic perturbation inference. And a DRVI, where we do this disentanglement of the latent spaces, thereby being able to interpret them. Both are not yet published, but we hope to bring them out soon. Challenges, as I was mentioned before, is this virtual cell. Of course, this could be sort of a digital twin, like how much constraint do we actually want to build in? I think those old purely first principle based ones kind of didn't work. So we need to have them data-based, but maybe there's a room for these additional constraints. All of this needs more benchmarks. And in the end, this will get really large. So how do we work on more than 1 billion cells? I think is a big challenge. And I think I was mentioning already this lab in the loop as one of the sort of key applications where you would go. I think I acknowledged already all the contributors and partners. So I just want to finish by showing my lab. I'm really acknowledging this fantastic team, grateful for funding partners, and I thank you very much for your attention.